Ebola virus. Ebola virus causes Ebola virus disease. This is very dangerous and often fatal one for 25–90% of patients. The virus was identified in 1976 near the Ebola River in what is now the Democratic Republic of the Congo, Africa. Ebola has cell membrane and it has nuclear proteins inside itself. Polymerase, which helps with replication. It has an outer glycoprotein. The genetic material, which is RNA, is within the nuclear protein. VP35 and VP40, which helps with viral replication. Ebola virus has a distinctive shape that resembles a long, thin, and flexible thread or filament, described as curved or looped structure. The virus is approximately 80 nanometers in diameter. Length can reach up to 14,000 nanometers. The filamentous morphology is a key characteristic of the virus, which belongs to the family of Filaridae. Ebola virus is a part of the Filaridae family. All Filaridae viruses has risk for pathogens. These pathogens are transmitted to humans through animals and have a lipid membrane. They have single-stranded negative sense RNA. All members of the Ebola virus can affect humans except for Bombali and Reston virus, which are highly pathogenic to non-humans. The transmission of the Ebola virus to humans happens through infected animals, such as fruit bats. Fruit bats carry the Ebola virus, and then the Ebola virus can be introduced to humans through close contact with the fruit bats, blood, secretion, or bodily fluids. Ebola spreads in the community through human to human. Let's have a quick look at how the Ebola virus can replicate or can infect a human. Here we're looking at the immune cell. An immune cell is important because immune cells can defend against viruses, infections, and other foreign bodies. The Ebola virus can be eaten up by the immune cells. Within the immune cell, the Ebola virus can somehow replicate. The immune cell is now infected, and as a result, the immune cell will secrete inflammatory molecules. Infection process. Infection occurs when a viral spike made of a glycoprotein located on the envelope attached to the host cell a glycoprotein receptor sites. Then the viral genome is released to the host cell. The virus replicates using the host cell's ribosomes to produce proteins during cell replication. Ebola virus disables the immune system by targeting macrophages and dendritic cells, infecting the liver, spleen, and other tissues, causing organ failure. Ebola disease is a severe illness that can lead to high mortality rates, sometimes as high as 90% in certain outbreaks. Signs and symptoms. Early symptoms. Signs and symptoms appear 2/21 days after infection with Ebola virus. Ebola first presents with headache, fever, fatigue, dizziness, sore throat, and muscle pain and body rashes. Middle stage. High fever, massive bloody vomiting, diarrhea. Late symptoms. Damage to blood vessels causes severe internal hemorrhage as well as bleeding from mouth, eyes, and ears, and organ damage. Many survivors have long-lasting severe joint pain and eye complications, ranging from vision changes to inflammation leading to blindness, muscle pain, hearing loss, and abdominal pain.
Oh, sorry, you probably just hear me crack my knuckles. So for this presentation and this project, I chose human immunodeficiency virus. It's very meaningful to me. My first job at a college was in an HIV clinic, and I decided through that I wanted to pursue healthcare. So yeah, very meaningful to me, and it was cool to learn more about it. So let's go into it. HIV was first isolated on February 4th, 1983. And this picture down here is actually a picture that was taken by the microscopist at the Institut Pasteur in France. And that was where HIV was first officially isolated. It is transmitted by unprotected sex from mother to child and through the use of contaminated needles. It is estimated that 84.2 million people have been infected with HIV and 40.1 million people have died since the beginning of the epidemic. Through this presentation, I'm going to go through the characteristics and relatives of HIV, but then I'm also going to talk about the pathological interactions that it has with the human body. I didn't find any instances of symbiosis. It throws off the balance of the microbiota, and untreated row of this virus leads to comorbidities and death. So I called that pathological myself, not myself. Anyways. So HIV is spherically shaped. It is about, you can see the envelope right there in the gray, is 0.1 to 0.15 micrometers in diameter. There are 72 glycoproteins, which are the green, and the layer of black dots is known as the protein matrix. And within this capsid in pink, there are two ssRNA molecules. And HIV is a part of a large family of viruses known as Retroviridae, and it is a part of a subfamily under that family known as Orthoretrovirinae. Some close relatives are HTLV1 and 2, and RSV and MLV. So to go into the interactions with the host, transmission occurs through body fluids such as semen, vaginal fluids, and blood. HIV will initially target the CD4 T cells. It will fuse to the host cell, uncode, and from that, HIV's RNA is reverse transcribed into DNA, and that DNA is inserted into the host genome. So just going through some like little stats right here, it's detectable, the RNA, after several days. It can be detected in the blood after 10. And the antibodies, which is normally the test that will diagnose people, are detectable after 30 days. And this is like the peak after like 30 days of exponential growth. So let's see, going back to right here at the second little bullet point, HIV is a virus. It doesn't do metabolism on its own, and so it relies on the CD4 T cells to metabolize and provide them, like do the glycolysis for them. So what happens is like there will be like an immense increase of glycolic activity in the T cells, and this gets them strung out. They are left vulnerable and susceptible to virus-induced death. And what happens from that is the CD4 T cell count goes way down, and at the onset of AIDS, it's about 200 like cells per millimeter. So if you're curious exactly, like I've heard about like, you know, AIDS and immunity, but now like having this like science and like knowledge, it's a lot more vivid. So let's go move on. So I did like look at the relationship between HIV and commensal bacteria, but it kind of is hard to tell based off the research. Like the research is unsure whether this is from HIV interacting with the commensal bacteria in the body or if it's the use of the highly active antiretroviral therapy. But regardless, there is a change in the composition of commensal bacteria throughout the body, but I focus on the gut and the mouth. HIV also will lead to cardiovascular disease, bone disease, renal disease, hepatic dysfunction. This is because of like the depleting like T cell count, and also inflammation that's caused. So basically, I want to end with the fact that World AIDS Day is on December 1st. And it's really important to keep this research going as well as like access to resources because a lot of people throughout the world don't have access to the antiretroviral therapy, unfortunately. So really just like working till that is good. And I'm really happy with like how far the science has gone.
Good afternoon, everyone. This is Megan Sorensen, and I'm going to tell you a little bit about the ubiquitous, the not so quite elusive rhinovirus. And I'm showing you this picture because this was actually me just this past summer when I was really, really sick on a vacation to Victoria, Canada. That was about as happy of a face as I could muster, but it was pretty grueling, and I just felt like garbage. I had a stuffy nose, a sore throat, a headache. And essentially, what I was experiencing was what we colloquially know as the common cold or the rhinovirus. So, the rhinovirus causes, it's kind of understood it causes the majority of what we know as the common cold cases, although there are other viruses that can cause them. But today we're focusing on the rhinovirus. And here are some fun facts. The rhinovirus is from the kingdom Ribovaria, the family Picornaviridae, the genus Rhinovirus, and it's an icosahedral shape. And actually, there are over 160 serotypes or strains of the rhinovirus. And this is due to the antigen arrangement on the surface of the virus. There's so many different variations of it. This is actually part of what makes it so hard for us to figure out how to vaccinate against the rhinovirus. But some more fun facts about the rhinovirus. It's a non-enveloped, positive sense, single-stranded RNA virus of approximately 7,200 base pairs in its genome. And the viral genome encodes for a single polypeptide protein, and actually proteases cleave it into 4 different viral proteins labeled VP1, VP2, VP3, and VP4. And these are what make the capsid and also what anchor the capsid to the genome. And actually, super fun fact, the rhinovirus is acid sensitive, meaning it's less stable in acidic environments. And this is in part explains why rhinoviruses seem to infect our noses and our upper respiratory tract a lot more than, say, like it's not going to like infect our stomach. And it's also closely related to the poliovirus. Even though they're in different genera, they are both under the Picornaviridae family, and they're both positive sense single-stranded RNA viruses. So how is the rhinovirus transmitted and how is it replicated? Well, I'm going to tell you. First one is droplet. Gross. I think we all know what that is, but that's little viral particles coming out when a person coughs or sneezes in your vicinity. And another way is fomites, door handles, utensils, pens, the gas pump, which I use a lot of hand sanitizer after I pump my gas. But these are 2 of the biggest ways that rhinovirus is transmitted. And the way that it's replicated is actually pretty interesting. It's a typical lytic cycle. But when rhinovirus attaches to the ICAM or intracellular adhesion molecule, which is on the surface of a host cell, it creates a conformational change that allows the virus to penetrate the host cell. And then a further drop in pH actually causes the capsid to uncoat, and it releases the viral genome where it can immediately hijack the host's ribosomes in that metabolic pathway to start replicating itself. And because it's positive sense rhinovirus, it can actually go straight to translation because its genome actually acts as mRNA. So it goes straight to translation, replication, and then it assembles its new little polyprotein parts into new viral genomes. And then eventually, when the cell lyses, all of these copies get released into the host, and then the cycle starts all over again. Some interesting, notable interactions that rhinovirus has with microorganisms. There's actually some good with the bad, but I'll start with the bad, or not so great. M. catarrhalis, which is a bacterium that's often found in the respiratory tracts of children, doesn't usually cause much harm or damage to the epithelial cells in the children. But when there's a co-infection with rhinovirus actually increase the host cell's ability to associate with M. catarrhalis, which cause more epithelial damage and more inflammatory response. But on the kind of great side, a study recently showed that in co-infection with SARS-CoV-2 or COVID-19 and rhinovirus, the rhinovirus actually triggered an interferon response that made most of the host cells non-permissible to COVID-19 attachment. And actually, in some cases, they showed that a rhinovirus infection could prevent COVID-19 from replicating within that host. So how do you protect yourself from rhinovirus, since it seems like it's everywhere and it can just make so many copies of itself? No surprise, hand washing. Hand washing is numero uno, with hand sanitizer coming in a close second if you can't hand wash, but always hand wash first. And the CDC actually has a really great page on their website all about quality hand washing and how you can protect yourself from getting a cold on your next vacation, just like I did. Thanks so much, guys, for watching, and look forward to seeing all of your videos.
Presenting on human papillomavirus for my Microbiology 234 term project. A little introduction. HPV. Nobody wants it, but many people have it. Human papillomavirus. It is a sexually transmitted viral infection spread by skin-to-skin contact that is quite common. It can cause warts, cancer, or no symptoms at all. There are 100 plus types of HPV. I found a lot of variation, somewhere between 100 and 200 strains, depending on the source and how they categorize them. It affects the body, as you see in the picture, with the different cancers, warts on the body. Shape and anatomy. It's pretty small for a virus, 52 to 55 nanometers. It's non-enveloped, icosahedral, single double-stranded DNA molecule with 8,000 base pairs bound to cellular histones, contained in a protein capsid composed of 72 pentameric capsomeres. Check out the image for a little more detail. Phylogeny. Papillomaviridae family, as I said, over 100 species of papillomaviruses. They break it down into 5 genres: alpha, beta, gamma, mu, and nu. There are different types that infect mammals, but they also infect birds, snakes, turtles, and fish. It's not known to have species-to-species transfer, though. There are 40 types that impact the genital tract specifically. Genital HPVs are closely associated with cervical cancers, and then they're grouped into high risk and low risk. 2 strains for cervical cancer are 16 and 18. Looking at how it enters the body, it enters through intimate skin-to-skin contact. That virus enters the epithelium through disruptions to the skin and or mucosa, and it infects basal stem cells. Some examples of skin or mucosa membranes where HPV can be transmitted are the mouth, vagina, or anus. Now, it is a virus, so it's acellular and doesn't have its own metabolism or growth, but it does rely on its host cell and manipulates their metabolism and growth. Whatever the means by which viruses act as pathogens, the infection results in changes in metabolism in the cells they infect and also in multiple cell types of the host that react to the infection, from Sumbria 2021. HPV causes changes in human glycogen, lipid metabolism, and other methods of metabolism that are correlated with traits that are common in cancer cells. Interactions with microorganisms. HPV is a parasitic virus that relies on its host cell. Research states host genetic variants in genes involved in human immune response may influence individual susceptibility to the long persistence of the virus and ultimately could influence HPV-induced cancers or tumor progressions, from Wakabayashi 2019. In other words, if you have their conditions or genetic disadvantages, you may be more prone to a progression of HPV in the body. Another interesting factor about interactions with microorganisms is the symbiosis with certain microorganisms, specifically of the cervicovaginal microbiome. And you can read those in the slide. Some of them, like Fusobacterium, are considered potential biomarkers for HPV-positive cervical cancers. It's postulated that dynamic interaction between HPV and the cervicovaginal microbiome creates an imbalanced cervicovaginal microenvironment that triggers the dysbiosis, enhances HPV persistence, and promotes cervical carcinogenesis. Nguyen, 2023. This research article also has information about potentially using probiotics in the way that you would treat cervical cancer or potentially improve your microbiome positive bacteria. And you can see in the picture an example of one probiotic. Interactions with other organisms. It's spread through intimate contact with other people. There are some environmental risk factors like smoking, having numerous sexual partners, or if you start having intercourse at an early age. Lastly, some good news is there are vaccines available since 2006. The CDC explains children ages 11 to 12 years should get 2 doses of HPV vaccine given 6 to 12 months apart. HPV vaccines can be given starting at age 9. Only 2 doses are needed if the first dose was given before the 15th birthday. Otherwise, you might need 3. Gardasil 9 is one of the HPV vaccines distributed in the United States, and it protects against 9 HPV types, and 16 and 18, as I mentioned, are the ones associated with cervical cancer. Thank you for listening, and I hope you learned a bit more about HPV.
The dangers of Hep C. Oh no, looks like Sarah poked herself with an infected needle. It looks like Sarah isn't feeling too well. Let's look at her symptoms. My muscles are starting to ache. I feel fatigued, nauseous, and now I have a fever. What is happening? Looks like Sarah contracted Hep C. But what is Hep C? I am a viral infection that causes liver swelling known as inflammation, which leads to liver damage. Approximately 3% of the world population is infected with HCV. Every year, there are 3 to 4 million new cases. Although Sarah contracted HCV through a contaminated needle, there are other ways it can be transmitted, such as an intravenous drug abuse, birth control, sexual contact, etc. Now let's take a look at its anatomy and shape. HCV is a spherical envelope positive strand RNA virus that belongs to the Hep C virus group in the Flaviridae family, which also causes viruses like yellow fever, West Nile, and dengue. HCV particles are round and vary in size, usually between 40 and 80 nanometers. There are 7 main types of HCV and 84 known subtypes. They are often linked with fat molecules known as lipoproteins. Since the virus exhibits a high degree of genetic diversity, it complicates vaccine development and treatment strategies due to the fact that certain genotypes and subtypes respond differently to antibacterial therapies. Although HCV and other flavirus shares some structural and genetic features, they differ significantly in terms of their treatment, in terms of their transmission modes, host range, and diseases they cause. Now let's take a look into the relationship between the gut, its microbes, and the liver in connections to HCV. In this study conducted by Dr. Obvious, it aimed to understand how changes in metabolism and microbial activities in these organs reflect the progression of liver disease, which results in cirrhosis. This diagram represents the gut, its microbes, and its liver, which are interconnected in the body's metabolism of nutrients and byproducts. Nutrients and microbial products observed in the gut travel directly to the liver through the portal vein, where they are further processed, as you can see on the diagram to your left. Since the infection can damage the liver through the inflammation and fibrosis, this disrupts both human cell and microbial metabolism, affecting the liver's ability to process nutrients, especially fats. After Dr. Obvious's research, it was found that there was a reduced ability to metabolize fat for energy, mainly in the liver cells peroxysomes and mitochondria, which are responsible for energy production and handling antioxidants. The gut microbiota was also affected by worsening liver disease. It indicated fat production, decreased methane metabolism, and damage to its intestinal mucus lining. In summary, due to HCV being a virus, it does not have its own metabolic machinery or ability to carry cellular processes like nutrition and growing independently. It has an oblique intercellular, which means it must affect a host cell in order to replicate and reproduce. Some ways HCV plays many environmental factors other than it being transmitted through blood to blood is, let's start by breaking it down together. Down below, I have placed some following examples, such as waterborne transmission, healthcare and blood supply, and disease progression. In environments with poor sanitation or contaminated water supplies, the risk of bloodborne transmission may increase. There are higher rates of HCV through blood transfusions and organ transplants from affected donors. Some other factors include diet, alcohol consumption, and obesity, which can progress to HCV infection. Now let's continue to SARS Inside Health. We must have made it into the liver. We have made it into the liver. We must reach the liver cell and attach to the co-receptors on the cell surface. I must release my RNA, which will be translated into its mature peptides. Oh no, looks like HCV has found us. They will only continue to unite and form a replication complex on the endoplasmic reticulum. Sadly, looks like the new viral RNA has packaged into new viral particles, which only leaves the cell to infect more cells. We must attack. There's more of us, fools. Oh no, I feel myself getting bigger. Rushing to the hospital. Oh no, looks like you have a liver disease. But thanks to your early detection, we can understand the virus better and begin with treatment plan.
My name's Hillary, and I did my term project on RSV. RSV is a significant pathogen that primarily affects vulnerable populations, including infants, young children, and the elderly, causing acute lower respiratory tract infections. RSV is highly contagious and mainly spreads through respiratory droplets, making it particularly transmittable in crowded environments like daycare centers and nursing homes. RSV infection can cause mild cold-like symptoms to severe bronchitis and pneumonia, which can be life-threatening in susceptible populations. Effective management and prevention strategies are essential in preventing long-term respiratory issues, such as asthma that may result from RSV infection. RSV is a negative sense single-stranded RNA virus that belongs to the Phenovidia family. RSV has a diameter of 120 to 300 nm, a spherical or phylomorphic shape, a lipid envelope derived from the host cell membrane, and two surface glycoproteins, fusion F and attachment G, crucial for viral entry and infection. The virus produces 11 proteins that aid in viral replication and assembly and can lead to the formation of syncytia in infection respiratory epithelial cells, contributing to severe respiratory symptoms. RSV is highly variable in morphology and structure, with even the same viral strain exhibiting different filamentous forms that facilitate its spread and penetration deeper into the respiratory tract. RSV can disrupt the respiratory microbiome, leading to dysbiosis and increased colonization by potentially pathogenic bacteria, resulting in increased disease severity and secondary bacterial infections. RSV infection can lead to increased colonization by Streptococcus pneumoniae, a common cause of bacteria pneumonia, which can worsen disease severity and increase the risk of mortality. RSV infection can also interact with other viruses, such as influenza virus, to cause more severe respiratory disease. These interactions between RSV and other microorganisms highlight the complex nature of respiratory infections that need for a multifaceted approach to prevent and treat. RSV's growth and metabolism can be influenced by the environmental factors such as temperature and humidity. Low humidity can enhance the survival and transmission of RSV in the air, while high humidity can reduce the spread of RSV by decreasing the stability of the virus's particles in the air. RSV can interact with environmental pollution such as particulate matter, PM, and ozone, which can exacerbate respiratory symptoms and increase the risk of RSV infections. RSV can be detected in various environmental sources, including wastewater and river water samples, suggesting that the virus can persist in aquatic environments. RSV relies entirely on the host cell machinery for its survival and replication, and its replication is highly dependent on the availability of host factors such as lipids, amino acids, and energy sources. RSV's ability to invade the host immune response and establish persistent infections presents a significant challenge in developing effective vaccines and therapeutics. Effective management and prevention strategies such as good hygiene practices and isolation precautions are essential in preventing RSV transmission. Developing effective prevention and control strategies for RSV will require a deeper understanding of its interactions with other microorganisms in the respiratory tract and how these interactions impact disease outcomes. Understanding the symbiotic, pathological, and environmental impacts of RSV is critical in developing effective prevention and control strategies against this significant respiratory pathogen. Further research is needed to gain a comprehensive understanding of the virus's behavior, interactions with the environment, and its impact on human health. Thank you.
Hello everyone, my name is Thuzar, and today I'm going to talk about herpes simplex virus, which is also one of my least favorite microbes. HSV is a common and highly contagious pathogen divided into two main types, HSV1 oral herpes and HSV2 genital herpes. HSV1 typically causes cold sores and around the mouth, while HSV2 causes genital herpes. Transmission occurs through direct contact with infected bodily fluids or lesions, making it easily spread through kissing, sexual contact, or even sharing utensils. So after initial infection, HSV becomes latent in sensory neurons. Reactivation can be triggered by factors like stress, UV lights, and immunosuppression, causing recurrent outbreaks. HSV virion is spherical, about 150 to 200 nanometers in diameter, and it also contains linear double-stranded DNA, capsid, teguments, and enveloped. It belongs to Herpes viridae family, and its subfamily is Alpha Herpes virinae. HSV is closely related with varicella zoster virus, which causes chickenpox and shingles, cytomegalovirus, which affect immunocompromised individuals and newborns, and Epstein-Barr virus, which is linked to infectious mononucleosis and certain cancer. HSV is an obligate intracellular parasite, meaning it cannot replicate outside of the host cell. HSV relies entirely on the host cell metabolic machinery to support its life cycle. So since HSV does not possess its own metabolic pathway for energy production or nutrient synthesis, it instead manipulates the host cell metabolism to create an environment conducive to viral replication. This includes upregulating nucleotide synthesis for viral DNA replication and modifying the host protein synthesis machinery to prioritize viral protein production. HSV also influences glucose metabolism, increasing glycolysis to meet the heightened energy demand for viral replication. HSV also has interaction with other microorganisms. Among them, one significant interaction occurs with human immunodeficiency virus HIV. So individuals co-infected with HSV and HIV experience more frequent and severe HSV outbreaks, and the presence of HSV can increase HIV viral load and the risk of HIV transmission. HSV can also interact with bacterial infection. HSV lesion can facilitate secondary bacterial infection, for example, Staphylococcus aureus. Moreover, HSV infection can disrupt oral and genital microbiome, potentially promoting conditions like bacterial vaginosis. HSV also have interaction with other organisms and the environment as well. In human hosts, it infects epithelial cells and neurons, establishes latency in sensory ganglia. It also inhibits antigen presentation and interferes with host antiviral responses. However, it does not survive well outside the host because it is very sensitive to drying detergents and disinfectants. It makes significant psychological and social impact due to visible lesions into high prevalence worldwide. That's why public health focus on reducing transmission through practices like safe sex practices and antiviral treatments. HSV also cause very severe diseases like hepatic keratitis, encephalitis, and neonatal herpes. So in summary, HSV remains a significant and enduring challenge in the field of infectious diseases. With its ability to establish lifelong latency and cause recurrent outbreaks, HSV continues to affect millions worldwide, impacting both physical health and quality of life. Its interaction with other pathogens like HIV and the potential for severe complications such as encephalitis and neonatal herpes underscore its complex role in human health. Therefore, understanding this HSV biology, transmission dynamics, and interaction with the host is very crucial for developing better treatments and preventative measures. Since science progresses, I hope that the more effective ways to manage and potentially eradicate HSV infection will be discovered very soon that reduce a significant burden on individual and healthcare system globally. So this is the works cited page which are the sources that I used to mention the information above. This is all for my presentation. Thank you so much for listening and your attention. Have a great day.
Poliovirus is the cause of poliomyelitis or polio, a disease ranging from asymptomatic infections to severe paralysis. There are 3 types of poliovirus, all of which can cause disease. Thanks to immunization, types 2 and 3 have been eradicated globally. Poliovirus is a spherical in shape and extremely small, with a diameter of just 25 nanometers. Its structure exhibits icosahedral symmetry, meaning its outer shell or capsid is arranged like a geometric puzzle. This capsid serves 2 key roles. It protects the virus's single-stranded RNA genome and facilitates attachment to host cells. Additionally, the lack of an outer envelope makes poliovirus highly resilient to environmental conditions like heat and pH changes, allowing it to survive for weeks in water or sewage. The virus is classified as an enterovirus within the Picornaviridae family. It is a single-stranded RNA virus, meaning its RNA can directly serve as messenger RNA in host cells. Its closest microbial relatives include other enteroviruses like Coxsackie viruses and ecoviruses, which also target the gastrointestinal system before spreading elsewhere. Like all viruses, poliovirus is an obligate intracellular parasite, meaning it does not have its own metabolic machinery. Instead, it hijacks the host cell systems to replicate. Upon infecting a host cell, it uses ribosomes, enzymes, and energy from the host to synthesize viral components. Poliovirus does not consume nutrients in the traditional sense. Instead, its replication depends on accessing the host cell's nutrients and molecular machinery. After entering a host cell, the virus rapidly multiplies, producing thousands of new virions that are released when the cell dies. Within the human gut, poliovirus may interact with the microbiota, which can either hinder or facilitate its replication. Studies suggest that gut bacteria can enhance the stability of poliovirus particles, potentially aiding in transmission. Poliovirus spreads primarily via the fecal-oral route, thriving in areas with poor sanitation. Once ingested, it initially replicates in the throat and intestinal lining before potentially invading the nervous system, leading to paralysis in some cases. It has a remarkable ability to persist in water or sewage, increasing its environmental transmission potential. Pathologically, poliovirus is infamous for causing paralytic polio. In severe cases, it damages motor neurons in the spinal cord, leading to irreversible paralysis or death. However, vaccines developed in the 20th century have been game changers. Thanks to these vaccines, global polio cases have plummeted by over 99%, though challenges persist in eradicating it completely. To conclude, poliovirus is a compelling example of how microorganisms can impact human health and society profoundly. While it once represented one of the most feared diseases, advances in microbiology and immunology have brought us closer to eradicating it entirely. Understanding its biology and interaction underscores the importance of continued scientific research and public health efforts. Here's my works cited. Thank you.